Hi, welcome back to LPHumananatomika.
The second component of the cell is nucleus. Nucleus is more or less a spherical mass situated within the cytoplasm near the center of the cell. It is a membrane-bound organelle which is around 6 micron meter in diameter, found in eukaryotic cells. It occupies about 10% of total cell volume. The nucleus consists of nuclear envelope or nuclear membrane, chromatin threads in a resting cell or chromosomes in a dividing cell, nucleolus, and nuclear sap. The contents of the nucleus are held in the nucleoplasm, similar to the cytoplasm in the rest of the cell. The fluid component of this is termed the nucleosol, similar to cytosol in the cytoplasm. In most types of granulocyte, a white blood cell, the nucleus, is lobated and can be bilobed, trilobed, or multilobed. When a cell dies, the nucleus becomes pycnotic with shrinkage and presents homogeneous hyperchromatin mass. The nucleus is stained with basic dyes since it contains abundant DNA and a small amount of RNA. Let us discuss in detail all the parts. Nuclear envelope, otherwise known as nuclear membrane, covers the nucleus. It separates the cell's genetic material from the surrounding cytoplasm, serving as a barrier to prevent macromolecules from diffusing freely between the nucleoplasm and the cytoplasm. It consists of two unit membranes, outer nuclear membrane and inner nuclear membranes, separated by a narrow perinuclear cistern or space which is about 10 to 50 nanometers. The outer nuclear membrane is continuous with the membrane of the rough endoplasmic reticulum. The outer nuclear membrane and rough endoplasmic reticulum are similarly studded with ribosomes. The inner membrane is a separate entity and ribosome free. It gives attachment to the ends of chromosomes and a dense coating of chromatin during interphase. Numerous nuclear pores are present in the nuclear membrane. A typical nucleus presents about 3000 to 4000 pores. The perinuclear space between the membranes is continuous with the lumen of rough endoplasmic reticulum. When seen from surface view, each nuclear pore is octagonal in shape and 80 nanometers in diameter. When seen in sectional view, it is funnel shaped where the outer end is narrower than the inner end. The outer nuclear membrane and the inner nuclear membrane fuse to form the nuclear pore. Nuclear pores are composed of multiple proteins collectively referred to as nucleoporins. It acts as a diaphragm for nuclear cytoplasmic exchange. Through these pores, mRNA, rRNA, tRNA are transmitted from the nucleus to the cytoplasm, but destructive cytoplasmic organelles like lysosomes are prevented from entering the nucleus. This size selectively allows the passage of small water-soluble molecules while preventing larger molecules such as nucleic acids and large proteins from inappropriately entering or exiting the nucleus. In the interphase, DNA material is present in the form of thin thread-like structures called chromatin and cannot be distinguished between each other. During cell division, each chromatin material starts coiling and condenses to its maximum and becomes thicker, shorter to form individual chromosomes and can be distinguished. The chromosome number is constant in a given species. In humans, there are 46 chromosomes. The details of the chromosomes will be dealt under genetics section. Nucleolus is highly refractile spherical or round body, but they are not surrounded by a covering membrane and is situated or located inside the nucleus. The nucleolus takes up around 25% of the volume of the nucleus. It is a compressed mass of a mixture of RNA granules and proteins. The synthesis of nucleolar RNA is regulated by the genes located in the secondary constructions called satellite bodies, that is, the nucleolar organizing regions in the short arms of the acrocentric chromosomes. The RNA is liberated from the nucleolus through the nuclear pores and appears in the cytoplasm. The nucleolus disappears during the prophase and reappears during telophase of cell division. The main function of nucleolus is to make ribosomal subunits from proteins and ribosomal RNA, also known as ribosomal RNA or rRNA. The nuclear sap is also called as nucleoplasm or karyoplasm. It fills up the interspaces between the chromatin threads and the nuclear membrane. It is a fluid containing proteins, enzymes, dissolved salts and several organic molecules. It serves as a medium for the transport of ribosomal RNA and messenger RNA to the nuclear pores. Nuclear sap reserves as a medium for the transport of ribosomal RNA and messenger RNA up to the nuclear pores. In addition, the nucleoplasm helps in cushioning and thus in turn protect the nucleolus and chromosomes while helping maintain the general shape of the nucleus. Coming to the overall functions of nucleus, it helps in protein synthesis, growth, cell division and differentiation. It stores the hereditary characters or DNA of the organism. It is the site for transcription. It stores proteins and RNA. It produces ribosomes, rewrite ribosomal RNA and proteins and helps in cell signaling.
If you like the video, hit the like button. If you have not yet subscribed, subscribe to the channel and hit notification button. Join my private Facebook community where like-minded students will support, discuss, encourage and push each other towards reaching the goal. For link, visit the description area. Thank you for watching.